And perforin, it turns out, works very similarly to the membrane attack complex. It will make a donut hole in the membrane of the cell lacking a major histocompatibility complex number 1. Here's another diagram. Here's the showing this. And now this cell is actually a cytotoxic killer T cell. This also uses perforin and granzyme. But I want to show you what perforin and granzyme do, because both natural killer cells and cytotoxic killer T cells have the ability to secrete these two molecules. So perforin is going to make these donut holes in the membrane, and then granzyme will be secreted. It can actually pass through these donut holes in the membrane or pores, and then it's a protease that basically can destroy proteins in the cell, and it can also lead to apoptosis or cell death. And that's what we're seeing here. The granzymes are going to trigger apoptosis or cell death in that cell. So whereas the natural killer cells can basically attack cells that lack MHC1, cytotoxic killer T cells have a different process. So the T cell receptor of a cytotoxic killer T cell has the ability to see peptides presented by an MHC1 molecule. So I want to remind you that MHC1 presents peptides from the cytoplasm. If there's no microorganism found in the cytoplasm of the cell, it'll be presenting self-peptides and everything should be fine. However, in a virus-infected cell, such as this one, this peptide in the MHC1 could be a viral peptide. So if a viral peptide is presented and a T cell with a CD8 receptor has the ability, so the cytotoxic killer T cells have a T cell receptor that can only see what is an MHC1. So MHC1 will present a foreign peptide, a 9 amino acid peptide, or a self-peptide. So if it's a self-peptide, it's going to be fine. If it's presenting a foreign peptide that's in the cytoplasm, then that could be a problem for this cell. Each T cell receptor on each individual T cell has the ability to bind and see one 9 amino acid epitope. So in this diagram here, for this cytotoxic killer T cell to kill this cell, virally infected cell, the viral peptide here needs to be an exact match for the T cell receptor, the CD8 T cell receptor. This has to interact in a lock and key mechanism. So if this viral peptide, 9 amino acid peptide, is the one recognized by the CD8 T cell receptor, well, T cell receptor plus the CD8 co-receptor, then it will then recognize it, and that will lead to the production of perforin and then granzyme to kill this virally infected cell. So interferon produced by the innate immune system can lead to the protection of neighboring cells. But that cell that was first infected by the virus is still a problem. It's the virus factory. So what happens is, as this cell is producing viral particles, the viral peptides will be presented on MHC1 on their surface, and then T cells, cytotoxic killer T cells, will go and sample the antigens presented on the MHC1. And if they see the antigen, the one antigen that they have evolved to detect, it'll secrete perforin and granzyme to kill that virally infected cell, and then it will not be producing any more viral particles. What kind of T cells can see the peptide presented in an MHC class II molecule? So here's a macrophage, which is a phagocytic cell. They're the only cells having MHC2, along with B cells, right? So the B cells and macrophages are phagocytic. Only phagocytic cells can have MHC2 and present peptides from phagolysosome fusion. MHC2 is always going to be presenting a foreign peptide. In contrast, the MHC1 usually presents a self-peptide from the cytoplasm, but if there's a cytoplasmic microorganism like a virus, it'll present viral peptides, where even some bacteria live intracellularly as well in the cytoplasm during an infection. Okay, so here's MHC2 presenting a foreign peptide from a phagolysosome fusion. Here is a CD4 helper T cell, and the CD4 will interact with the T cell receptor for it to see and check out the antigen presented in an MHC class II molecule. And every helper T cell has the ability to see and interact in a lock and key mechanism with one 9-amino acid epitope antigen. This is a very, very specific reaction. And if this reaction is happening with this 9-amino acid foreign antigen presented, detected by the CD4 T cell receptor of a helper T cell, then this T cell will become activated and start replicating to produce more of this T cell. And likewise, after this cell has killed the infected cell with perforating granzyme, this T cell, cytotoxic killer T cell, will replicate to make more of them because there's likely other cells infected by that virus, right? Because that cell has released many viral particles that have now infected new cells in the body. So we need to amplify this cytotoxic killer T cell to have more of them to go kill other virally infected cells in the body. And the helper T cell, which we see here, this is going to play a role in helping B cells to make antibodies. And we're going to go through that. So I want to now show you how this can work. So here's a person that has been infected by a microorganism. So we have B cells that have antibodies on their cell surface. Every B cell has antibodies that can recognize one and only one 9-amino acid epitope. Here we see the antigen. When a B cell binds to its antigen, it'll internalize the microorganism attached to that antigen via phagolysosome fusion, and we're going to go through that process in greater detail. Okay, in a minute. But basically, this B cell, once it finds its antigen, it's going to need its partner T cell that binds that same antigen to come and find it. So in parallel, we need a B cell to find the antigen, and then we need the T cell to see the antigen inside an MHC2, the helper T cell of a macrophage, for example. It needs to become activated right after seeing its antigen on an MHC class II. It becomes activated, it replicates, then it goes to look for its partner B cell. When they find each other, then the B cell can differentiate into a plasma B cell and secrete antibodies. And now I want to show you, review what I told you about the role of the CD8 cytotoxic killer T cell. The cytotoxic killer T cell does not play a role in antibody production. Instead, it actually directly kills the virally infected cell. So here we have the MHC1 presenting a cytoplasmic peptide of a virus. Then it's going to be an exact match for the T cell CD8 killer T cell receptor. It'll basically see the antigen and then secrete perforating granzyme to kill the cell. And as it is activated, it'll also replicate and begin to replicate to make divide to make thousands of that T cell, so it can find other virally infected cells in the body. What I want to show you here is the process of the B cell becoming activated. Okay, so we have a B cell that is going to bind an antigen of the microorganism, and critically, B cells can only see what is on the surface. They cannot see what's in the middle of a microorganism. They can only see what is on the surface. And I want to point out that B cells are amazing because they help our immune system only make antibodies to surface structures. And this is so important because an antibody for example, RNA polymerase of a bacteria that is inside the cytoplasm is going to be useless. The B cells play the absolutely critical role of ensuring the antibodies made by our body are going to be antibodies to surface structures of microorganisms. So this is absolutely so critical that they play this role in the immune system. So the B cell will find the antigen attached to the microorganism. It will then phagocytose it, bring it into the phagolysosome, and then it will present that same 9-amino acid epitope that its antibodies bind. It'll present that on an MHC class II protein. That needs to happen. Another thing that needs to happen is for a phagocytic cell, like a macrophage, to basically phagocytose the microorganism, bring it into phagolysosome fusion, then present the 9 amino acid peptides on an MHC2 molecule. That 9 amino acid peptide is going to be there. T cells are antigen samplers. Okay, so this CD4 helper T cell is going to sample the antigens in the MHC2 molecules to see if it fits with that antigen in a lock and key mechanism. It has to be an exact match. If it's an exact match, the T cell is going to be activated and proliferate and make more, like thousands of this exact T cell. They are going to go to the bars of the body, which I call the lymph nodes, to see if they can find their partner B cell. And at that time, they need the B cell to have come into contact with the same microorganism so that they can then make the B cell differentiate. Okay, so here is a CD4 helper T cell. The T cell receptor of a CD4 helper T cell can only see what is an MHC2. In contrast, a cytotoxic CD8 killer T cell can only see peptides presented in MHC1. There's no cross-talking at all. So the amazing thing is that if this 9 amino acid epitope presented on the B cell is an exact match for the peptide that the T cell receptor evolved to bind, then they will secrete cytokines to stimulate differentiation, making the B cell into a plasma B cell, and the plasma B cell will create about 2,000 antibodies per second. And I think of the plasma B cell as a pregnant B cell because it's giving birth to all these antibodies. After this process has occurred, where the soulmates, I think of the B cell and a T cell as a soulmate, right? The helper T cell only can bind that one 9 amino acid antigen. The antibodies in the B cell can also only bind that one 9 amino acid antigen, and they're in this huge human body, and basically they have found each other. So when they found each other, which is the point of their life as a cell, is to the B cells and T cells want to find each other so they can make antibodies to get rid of infection. Then memory B cells and T cells are created, and those can then make antibodies really quickly and also in high amount. So the reason that the body makes it cells go through many steps to be creating antibodies is that we want to prevent autoimmunity. It shouldn't be easy to make antibodies, right? So because antibodies can cause great destruction in our body if they react with a self-peptide. So our body goes to these great lengths to make sure that you need a macrophage to be phagocytosing the microorganism, right? And then to activate a T cell. You need a B cell to come into contact with that same microorganism then that this B cell evolved to detect the antigen of, and then present it on MHC2 to an activated helper T cell, and then for those two cells to stimulate each other to make a plasma B cell that'll secure all the antibodies, and also a memory B cell and a memory T cell. I should also mention that when the B cell internalizes this antigen, it also becomes activated. So the T cell becomes activated by seeing its antigen. The B cell also becomes activated and replicates in the thousands when it finds its antigen. So the activated B cells can live for about a week. It can go to the spleen and lymph tissue, right? And the lymph nodes are like the bars of the body, right? Where all these different kinds of white blood cells can come together. And only when the B cell finds this partner T cell can a plasma B cell created that produces 2,000 antibodies per second, and a memory B cell be created, and a memory T cell, which is not shown, but that memory T cell is made too, and that allows for the rapid production of antibodies in a secondary infection that can give immunity.